Hi, in this new tutorial, we will cover the tracking of moving objects like population of cells or other microorganisms. Before we dig into the analysis, let me remind you some critical parameters for the imaging of dynamic phenomena. The first thing to have in mind is a trade-off between resolution and volume of observation. While higher resolution objectives allow to better distinguish neighboring particles, they also have a smaller field of view and a narrower depth of focus, which result in a smaller volume of observation. As a result, less particles are visible in a given field of view, and the particles are more likely to escape the volume of observation during the time of acquisition. As a result, at high resolution, tracks tend to be shorter than at low resolution. On the other hand, if the resolution is too low, one cannot precisely quantify the displacement of the particle, nor precisely distinguish between neighboring particles. Keep in mind though, for fluorescent point emitters, subpixel localization is possible using localization algorithm like Gaussian flinting, for instance. The second key parameter when observing a dynamic phenomenon is the time resolution, or how often we should take a picture. Having more pictures allows to have a finer description of the dynamic phenomenon. However, it increases the size of the dataset. It increases photo bleaching if fluorescent samples are used. On the other hand, if too few pictures are used, one can possibly miss an interesting event. A trade-off should be found between the properties of the fluorophore and the timing of the dynamic event. To illustrate the impact of time resolution on tracking, let's consider the following illustration. You can see here two particles artificially colored in orange and red. In reality, when you perform tracking, all particles typically have the same color, so you cannot use this information to link them. Those particles are diffusing over time, and 3 positions were recorded. To link the position over time, in other words to reconstruct the track for a given particle, tracking algorithm will usually look for the closest particle in the image of the following time point. This is called the nearest neighbor search. If now we had reduced the time resolution such that only the first and third position of each particle would have been captured, the nearest neighbor search would have connected the wrong particles when reconstructing the tracks. This linking error might be obvious for a human, especially if the two objects have different aspects. However, in most cases, the algorithm relies only on current and past position of the particles to reconstruct their track. Therefore, in the second case, the time resolution is no more sufficient compared to the mobility of the particle to perform a reliable tracking. The number of time points should be increased. For more insight into practical consideration about tracking and time-lapse experiments, I highly recommend to consult this review paper. Once the data acquired, it is time for the analysis. Some pre-processing, like background subtraction or deconvolution, can actually help with fluorescent images before actually starting the tracking. Then there are 2 solutions, either manually tracking or using some kind of automated solution. If you have only a few particles, or if an automated solution is too challenging, manual tracking can offer very reliable results. Let me show you an example in Fiji using the manual tracking plugin. Before starting the tracking, it's important to set the pixel size and the time interval, which are used for the measure of velocity. Once this is set, you can click the Add Track and start clicking the center of the object progressively over time. You can see that for every click, a new line is added to the result table with information like the coordinate for the click position and the velocity of the particle. Once you're done tracking a particular object, you can end the track and start a new track. Finally, once all particles have been tracked, you can render the result of the manual tracking over the image. Eventually, you can also activate an option during tracking to correct the location of the click position automatically. This is particularly useful for bright round particles. If you have a high number of particles to track, a semi-automated approach might be worth the try. A popular and well-documented solution, available in Fiji and IC, is TrackMate. TrackMate offers a step-by-step guide to first identify the particles, link the position over time, and finally visualize and edit the tracks. If you need to do tracking in 3D, Mammoth, which combines the functionalities of TrackMate and the Big Data Viewer, is also available. Elastic also offers advanced functionalities for tracking. It combines the power of machine learning-based segmentation with tracking functionality in an interactive interface. It also provides dedicated solutions for the tracking of small animals like mice. DeepLabCut is a powerful solution that relies on deep learning for pose estimation. It allows to track multiple reference points on moving objects and specimens. It currently requires some Python programming skills, but it comes with dedicated Jupyter notebook to help you get started. Tools for the visualization of the tracking results are also provided. With this, I finished this lesson on tracking. As a summary, we have reviewed some critical parameters for the imaging of dynamic phenomena. We also have seen the nearest neighbor search algorithm used to link particles over time. And we also have seen some software solution that you can use to perform tracking and analyze the result. Thank you and goodbye.